So I'm going to move on to the next speaker, Shib Basu, who is going to make a 20-minute presentation, and then we'll have 5 minutes for questions. Dr. Basu has been involved with agricultural biotechnology research and development for many years. He has a lot of experience with enzyme technologies and has also worked a lot with microbial solutions to improving crop productivity. Dr. Basu is a senior scientist, microbial informatics and illumination at Indigo Agriculture based in Boston, USA. Currently, Dr. Basu is focusing on nominating microbes for improving resilience under abiotic and biotic stresses as indicated on the brochure that you sent earlier. Now, Dr. Basu is going to present on Indigo's microbial science and screening for microbes to improve plant productivity and fitness. So, Dr. Basu, the floor, so to speak, is yours. Thank you very much.
Thank you so much. Thank you for the organizer, the Department of Science and Innovation of South Africa, and also ICGB. And special thanks to Vittorio for actually inviting me for this colloquium. Okay, thank you so much, everyone. What I'll try to describe to you is Indigo's microbial science in particular. Okay, but before I go into that or dive into that scientific area, I'll also try to introduce Indigo as an ag company and as a system innovator in agriculture. Okay, so Indigo started in 2014, about 6 years ago, with a mission and actually provide solution to many of the things that Vittorio actually highlighted in his talk of the challenges that right now this planet is facing and the humanity is actually facing. So we looked our focus, well, the main motto or main mission is to harness the nature to help the farmer, and to do that is in a very sustainable way. Okay, our focus is to provide improvement, provide profitability, and reduce the risk for the farmers. Okay, and do that in a very sustainable way. So that's as our core mission. Okay. The way we see it, and secondly, also to align all the agricultural practices and agricultural solutions that the Indigo provides and help to improve the consumer health. Okay, so these are the three biggest things that profitability, to maintain the sustainability for the looking at our planet, and then also the ultimate consumers, okay, look at their health too. And you will see that the way we develop our product, we keep these three core ideas always working behind everything that we do. So as I say, the Indigo started into about 6 years ago. Okay, but it developed well. It grew pretty fast. Okay, right now it's still a startup. Okay, but we have now about 1,200 employees. And it started in Boston, which is our global headquarters in North America. Okay, and then we extended the commercial operation is directed from the Memphis, Tennessee, which is in the middle, almost in the middle of US. Okay, we have a research facility in Research Triangle Park in North Carolina. That's also in the US. Okay, we have our footprints and our South American headquarters as in Brazil, Sao Paulo, as well as in Argentina. In Europe, our European headquarters is in Basel, and from there we work with the different European countries. Okay, as far as any other country, so we have presence at least in 3 different continents. Okay, as well as we have in India, we have a joint venture with Mexico. Okay, we still do not have our presence in Africa, or we also don't have anything in Australia. Okay, but we hope to extend that too. Our main focus is 5 different crops. Okay, these are the main crops that we are working, because we cannot work with too many of them. We have to get focus on which we can handle. One is cotton, wheat, soybean, in corn, and rice. Okay. So the Indigo actually strives to be the first innovator in agriculture, and bring a holistic approach rather than a reductionist approach. Also, this is keeping in mind our mission of sustainability. Okay, we also recognize that in the plant, especially in the field, faces with multiple stress at a time. So a combination of product is often product and solutions are needed to take care of those stresses. Okay, and in order to do that, we build up a whole infrastructures, including in digital technologies, is different instruments, as well as different sensors to bring in new data straight from the field into the lab. Okay, we also have business models helping the farmer in different ways, is to increase the profitability. At the same time, we also have microbial solutions, and I will discuss more about that. Okay. So with this mission in mind, there are 4 different offerings from Indigo. One is carbon, okay, for a better planet and a better our farmers' bottom line. And Indigo's carbon, and suppose the farmers for their transition into a more beneficial practice. Okay, we pay the farmer for carbon sequestering. Indigo actually is the first company to purchase agricultural carbon on credit from the farmers. Indigo also have a marketplace where the farmer we provide the logistics and a framework for the farmers to sell their product. Okay, sell their grains. It is something like an Amazon of agriculture. Okay. Indigo has also extended into transport, which is called Indigo Transport. It is a simple and more profitable way to actually haul, providing efficient hauling service. You can regard that as the Uber in agriculture. It is actually a digital logistic platforms for connecting the shippers or the carriers of agricultural product and making the hauling as efficient and hassle-free for the farmers. Okay, and then of course we have the microbial products. Okay. Now, this brings to our microbial science, and in rest of the stuff, I'll be concentrated on the microbial science, and where microbial R&D and its screening pipeline. This is where I also work. Okay. So this is particular while diagram shows our microbial pipeline. Okay, it starts with the collection of samples, what we call focus searchings. Is very similar to what Vittorio actually described. Okay, and I'll describe it actually what we do also in soon. Once the microbes are isolated, they are of course always build up the whole collections of the microbes, as well as the whole genome sequencing, not only amplicons, but also a whole genome sequencing, and synthesize a hypothesis related to each one of the microbe, how they may benefit the plant, and nominate it into the screening platforms, where it goes through high throughput screening in the laboratories and the grow room, and then moves into the greenhouse, and finally the top performers from the greenhouse, and also all the information that we gather in the laboratory. It helps us to find that our best, and then try the field trials. And we also collaborate with our partners, with the farmers, where the large acres are also cultivated, and large acre trials are carried out. And so this is the whole pathway towards commercialization. And every point of whatever that means, we have different data collections process, and they finally again comes back to feed our data collections, as well as to build up or refine our hypothesis how the microbes can be beneficial to the plant. Okay. Okay, so to start with the focus sourcing part, okay, so we collect microbes from the field, okay, and we concentrate, okay, our microbes are collected from inside the healthy plant and not from the soil. We aim to collect the endophytes, which, as you can imagine, have, of course, also will be playing a vital role helping the crop survive under stress conditions, because the crop itself is their host or their home. And this helps them to motivate to ensure a healthy home for themselves. Okay, and we collect them, and we collect them and culture them, and then also build up our own genome, our own collections of that. Okay, in addition to that, we also we do amplicon on well, we definitely do a number of things with this each one of those microbes that we collect. Okay, among them, the important things are whole genome sequencing, taxonomic classifications, and other things. Okay, and this helps us, and also, like, well, the video actually described comparing between the controls and the winners. I'll describe a little more on that one too. So by this way, we have a collections of over 36,000 and different microbes, okay, both bacteria and fungi. As a comparison of a community sequencing of control, by which we means a stressed plant. Okay, so we collect a microbes from the stress. So we go into field which are stressed, okay, either by some abiotic stress or some biotic stress, and collect samples from both the healthy plant and the nearby stressed out plant. So the control here means actually the stressed plant, and the winner is the one that is surviving well even under the stress. And here you can see that there is a clear shift from the gamma to the beta proteobacters, as in case of the corn on plants which are stressed and under drought. So among the different kind of stress that we focus in our company, okay, and in our screening platform, these are the 5 different ones. Okay, 3 of them are abiotic stress and 2 abiotic stress. Among the abiotic stress, nutrient and use efficiency is one of the main focus, mainly nitrogen and phosphate to some extent. And second is the drought stress, and then the cold wet stress, which has a big impact, especially at the early phase of the plant and well, early phase of the plant at the seedlings. Okay. Among the abiotic stress is we focus mainly on the fungal stress, fungal pathogens, okay, and then of course the nematodes. So as I said, our pipeline starts with the first analysis of the genomic sequence, okay, among other things, okay. There are other criteria too, but one of the main thing we analyze is genomic sequence, and these slides actually describe some of those, okay. And here actually we screen in silico the genomic sequence, and we look for genetic features that are relevant to a microbial functional trait, and that can help the plant to improve their growth as well as the yield. Okay. And when we develop all this hypothesis for this, okay, we keep in mind all these different ways by which the plant can be benefited by the microbes. Okay, so I mean, just for the sake of the time, I have to go pretty fast over here. But if I am slowing down, please remind me. And if my time is over, please remind me. Okay. Once these hypotheses are developed for each of the microbe based on the genomic sequence, they are channeled into different assays, which is the stage 2 of the screening pipelines, where we look for different assays, and we have a host of different in vitro and in plant assay. Those are carried out in the laboratory as well as in the grow rooms. These assays are usually at the most 7 to 8 days long. We have assays for all the different 5 different crops of our interest, and also all different and stresses of our interest. In our assays at the stage 2, we collect data for multiple metrics. Okay, for example, in plant assay, we capture high resolution image scanning both the shoot and the roots. Okay, this shows, and this helps us actually compare the root and shoot morphology. In addition, we can also do hyperspectral scan, and our hyperspectral scan covers the whole range from 400 nanometers to 240 2400 nanometer, and this helps us actually to analyze both the health and other characteristic features that are changing as a result of microbial treatment. And here those different colors lines are coming out from different plants that are stressed with different amount of nitrogen level. So slight changes in the nutrient conditions can have a big impact, and that can be captured with the hyperspectral scan. So that was mainly the screening in the laboratory and the grow room, and then it moves into the greenhouses, is where the plants are actually grown from 3 weeks to the full maturity. And here also we look into the plant morphology, both above ground and below ground. We do tissue nutrient analysis. We do phenospec metrics. We capture the phenospec metrics as well as hyperspectral imaging at this stage. So the best performance are based from this, okay, are then assessed and then compared, and the top performers are actually sent into the field trial. I won't go into the details of the field trials, but as you can see in some of the selected pictures, as often we see a clear difference between in the indigo microbe treated plants and compared to the untreated one. And in multiple crops, in multiple field trials, over multiple years. So this kind of summarizes the whole pathway. Once we collect and the whole process from collecting the microbes to all the way to the commercializations takes about 3 years. Okay, we start with thousands, 10,000s of microbes collected from on the field to develop about now 3 to 5 microbes that are reliable and with high efficacy, and that can change and that can and provide benefits to the plants. Okay, of course, this is right now the current efficiency and current stage, but we are continuously working to improve the efficiency of this pipeline. Okay. Now I'll just give you a short descriptions of our portfolio right now. These are the different products for different crops. Okay. It highlights about 10 different products for corn, on soybean and cereals, mainly wheat. Okay, we are still working on the rice one, and this actually really addresses different kind of stress, both mostly the abiotic stress is right now. just to give you a flavor of our indigo product, I will just to give you one example and how it's performing. So this is this Indigo 206, okay, is a colonizer of the roots, and you can see here on the root surface as is the our microbe of 206, okay, and those are actually indicated by the stars, okay. Yeah, in the greenhouse, we can see a clear difference between the untreated and the indigo treated, and in particularly, you can see a 20% increase is in the leaf nutrient and uptake, both or nitrogen as well as potassium uptake has increased due to the indigo 206 treatment. Well, how does it helps in the uptake? Okay, we are definitely doing a lot of research to understand that. But one thing that came out to be very interesting is that it appears to be increasing the colonization of mycorrhiza in the that is present in the soil, and you can see a clear difference between in the untreated and mycorrhiza samples taken from 206 treated and roots of the infestations of the mycorrhiza. Okay, and you can clearly quantitate that. So this could be one of the mechanisms by which this microbes, there could be other mechanisms too, but this is something that appears very interesting. Not only it is it may be doing helping the plant to uptake by itself by colonizing the roots, but it also acting as a helper microbe for the mycorrhizas. And this is also can be seen in from samples collected from the field trials too. Okay, clearly there is a more mycorrhiza infestations or more mycorrhiza colonizations in indigo treated plant. And this shows us a summary of field trials carried out over 3 years with this Indigo 206 eggs in different multiple areas. These are in Texas, in Kansas, and in Oklahoma. And you can see an average increase of over 10% and yield in winter wheat can be observed over a period of 3 years under different kind of stresses and extreme weather conditions. Not only yield, but also, as I said, that one of our Indigo's objective is to improve human health, and this is another thing that I observe, and especially the grower observes, is that there is an increase in the protein content in the wheat that is isolated. So the protein content moved from 12.75 to 13.8 in the indigo treated in wheat. And so there's an increase of about 9% in the protein content. Okay. So this is just a short glimpse of what we are doing at Indigo. We are working towards to be successful by 2025, if definitely. This is what we hope for, and if we are successful, these are some of the things that we'll probably be able to achieve. The agriculture will know how to produce enough food for these growing populations. So we will actually give back to the farming community about $5 billion of wealth. Okay, and this comes from the carbon credit that Indigo is selling. And the farmers who uses Indigos will be able to use less fertilizers and less chemicals, 50% less fertilizer, 90% less chemicals, and this will make the farmers net carbon positive. And finally, of course, that the consumers they will be able to trust where they're from best is on our labeling and other stuff. Digital technologies is we'll be able to stress our plant Indigo product and able to provide a healthy and sustainable solutions to agriculture. Okay, that's all I have. I think I don't know about the timing. I did not keep it.
Yes, the timing is actually up. Are you done?
Okay. Yeah, I'm done. Thank you so much.
Thank you very much, Basu. Yeah, we have 2 minutes for questions. And so I'm just looking at the questions that have been sent by some of the participants, and I'll just go through one or two. So one of the questions is, a lot of this work is aimed at plant health. However, can one use inoculants to influence the chemistry of the plant? For example, nutrient properties or flavonoid content. Is that possible? I'll ask the two questions at the same time so you can just answer one time. So that's one question, whether the inoculants can be used to influence plant chemistry. And two, I think in your presentation, you may have talked about an Uber model. And there's someone who also just wants to know more about that model. And I'll ask a third question that has just come in. What species is the Indigo 206 inoculant? So those are three questions. One, inoculants to influence chemistry. Two, a bit more explanation about the Uber model. And three, the species of the Indigo 206 inoculant.
Okay. So for the first question, yes, as a scientist, I do believe that, yeah, the plant chemistry can be changed, and even things, important chemicals like flavonoids and other things can be changed. But we are not doing that kind of research right now within Indigos. That is not what we are testing right now. However, as I showed you that in case of 206, okay, 206, it is we were able to change the protein content and in wheat, okay. So once we look at the grain and we do the analysis, we do see changes happening. But we are just as a commercial company, okay, just as a company, we are not going into the details of too much into the chemistry, ecological change. About the strain, and I can give you that information. So, okay, this should be in the label. I don't. I should be able to. I can send it to you. Okay, that exact, because that means just for our own confidentiality, there are some restrictions, but I can give you all this information, I think. What was about the Uber? Okay, so about the Indigo Transport, okay, it's the same Uber model where we have a logistics system or platform where a farmer can go if he or she is want to transport the grain from one place to another. He can contact a carrier who can help him to move the grains. Okay, it's very similar to the Uber concept, but here we are reaching out the farmers. The farmers are the customers, as well as the people who have truck and people who have the carriers are the ones who are the Uber drivers. Okay, it's very similar concept. We are providing the logistic, and this is happening only in North America right now.
Okay.
Okay. Thank you so much. Thank you.
Okay. Thank you so much, Dr. Basu.